Today we're talking about Chapter 9, Sections 3 through 6, and this is going to be the different applications of biotechnology, particularly focusing on the fields of forensics, medicine, and agriculture. Our first topic is DNA fingerprinting. And the thing about DNA fingerprinting is that it's not an actual fingerprint like from your thumb if you leave behind a fingerprint at the scene of the crime. This is a fingerprint based on the pattern that your DNA would make. So make sure you're not confusing the two. DNA fingerprints work because if you look at the non-coding regions of DNA between individuals, there's a lot of differences there. The coding sections, the actual genes for making important proteins, those are mostly the same from one person to the next, but all the non-coding DNA in between accumulates differences over time. And so if you look at different individuals, they have different sequences in those non-coding regions. One particular thing that is really helpful in making a DNA fingerprint is that there are some DNA sequences that are repeated in our DNA. We call those short tandem repeats, and they can be repeated just a couple of times, or they could be repeated 7 or 8 times. And the number of repeats is going to differ from one individual to the next. So this is one of the major contributing factors for person A having one specific DNA sequence and person B would have a different one. So in this example here, these 2 pieces of DNA would represent the 2 alleles that a person would have. Being diploid, there's 2 copies of every chromosome. Well, whatever you inherit from your mom is going to be similar to whatever she had, and she inherited from her mother, and so on and so forth. And then you inherited your other one from your dad, and that can have a different version of the DNA on it, or a different allele. You can see dad's allele would have 6 repeats in this place, 7 repeats in this place. So every person has 2 different copies of those chromosomes that they inherited from 2 separate people. So that is 2 separate locations on the DNA that might differ from person A and person B. So you'll notice that based on the number of repeats, if we were to run that on a gel, something like 7 repeats, that's a much larger fragment, it would show up on the gel in this location, whereas the 3 repeats is a smaller fragment, it would show up closer to the bottom because those smaller fragments move faster through the gel. So comparing person A and person B here, just looking at these 2 chromosomes, we can see that the pattern produced by gel electrophoresis in person A looks very different from the pattern produced by person B. So that makes it very helpful to identify individuals using this process. So main uses for DNA fingerprinting is basically just for identification, either identification at a crime scene or like what you see here, identification of paternity. Now, we've done a lot of practice in class about identifying a suspect from a crime. That's really easy. You just look at the DNA sequence and you try to find an exact match. While we have this picture up here, though, let's talk about how it's a little bit different to use a DNA fingerprint to determine paternity, or who would be the father of a child. To do this, you have to look at the child's DNA fingerprint, and you have to look at that of the mother, and know that every band of DNA is either going to be an allele inherited from the mother or the father of this child. So if you look at each band, this one obviously corresponds to the band from the mother. This one is a little bit lower. It doesn't correspond to the mother, but it does exactly correspond to this father band over here. This one came from the father, this one came from the father, this one came from the father, this one from the mother, and so on and so forth. So if you were to look at every single band, you would find an exact match in either the mother or the father. So that would rule out this person right here as the father of this child, because let's just look at this band as an example. This band in the child is not present in the mother, and it's not present in this person, so we can immediately rule that person out as being the father of the child, because the child had to inherit this from one of his 2 parents. But since this person does have the band there and matches all the way down, we can determine this is the father of this particular child with that mother. Some examples of how we use DNA fingerprinting. We've talked about criminal cases and paternity testing. Also biodiversity. Biodiversity would be the diversity of gene sequences in a population. Particularly if you're studying endangered species, you want to make sure that there's still some diversity left in that population. You can identify illegal poaching, the elephant ivory tusk trade. You can actually extract DNA from confiscated tusks and figure out what population of elephants it came from. And then also you can use it to identify genetically modified crops. There are certain genes that are commonly used in all genetic modification, so you can identify that by creating a DNA fingerprint of different crops and comparing those sequences. Our next topic is going to be that of cloning. Now, a clone is simply a genetically identical copy of an organism. And this is something that we tend to think of happening in a lab, but it does actually occur in nature. Bacteria clone themselves through binary fission. Plants can clone themselves. They can send their roots out, and then another shoot will grow up far away from that original parent plant. Well, that would be a clone, and it's genetically identical to the original plant. Some animals can actually clone themselves. We've talked about this example. This is a hydra. And the bud coming off of the side, that's a clone of this original. It's completely genetically identical. And then there are cloned humans, but these are natural clones. Identical twins are a clone of one fertilized egg as it is developing very early on. If that simple ball of cells gets separated into two separate balls of cells, then each one of those has the potential to grow into a different individual. And that's how you get identical twins. Since they both come from one fertilized egg, they both have the same DNA sequence. Now, the cloning that we normally think of if we talk about lab cloning is called somatic cell nuclear transfer. This is a process where the name kind of tells you the gist of what is happening in the process. We have as an example here, if we're cloning a mouse, we're cloning this gray mouse right here. First thing we need to do is we need to get a donor egg cell. So we're using a brown mouse as the source of our donor egg. The donor egg is important because it has all of the cytoplasm with the correct chemicals and the correct organelles in it to be able to grow into an embryo. A somatic cell doesn't have the ability to grow into an embryo, so we need the egg cell for that. But the nucleus of the egg cell is going to be haploid. There's only half the number of chromosomes that you need to make a healthy mouse. So we need the DNA from a somatic cell. We take that from the mouse we're trying to clone. So all of the DNA of this clone that we're making is coming completely from this somatic cell of the gray mouse. You insert the somatic cell DNA into the egg cell, and then you stimulate that new cell to start dividing. You can do that using a chemical signal or even a little tiny electrical shock. Then this embryo or this ball of dividing cells, we can't just keep that in a lab and have it grow in the lab. We have to implant it into a surrogate. So this white mouse here would be our surrogate. The surrogate basically provides a place for an embryo to grow and develop, and then the surrogate will give birth to the clone. Now, the clone is not genetically related to the surrogate at all. The clone is a complete genetic copy of that original gray mouse that we took the somatic cell from. So that is the process of somatic cell nuclear transfer. That is the most common way to clone an animal in the lab. And in fact, that's how Dolly the sheep, who you can see pictured right here, that's how she was cloned. Dolly is a clone of another sheep. So Dolly herself was the clone. First successful cloning of a mammal, and that happened back in 1996. So the reasons for cloning could be things like to grow organs for transplantation. You could clone an animal model to study a disease. Animals are very commonly used as a way to study new treatments and drugs. You could clone to try to save an endangered species. If there's not very many members of a population left, you can clone those members of the population and increase the population size that way. Also, if you are a farmer and you have livestock, you could clone those prized livestock instead of trying to recreate that through selective breeding. Or there's even companies that will clone a pet for you. If your pet passes away and you want another copy of that same dog or cat, there are companies that will do that for you at a pretty steep price tag, but they will do it for you nonetheless. There's also a few concerns about cloning. Historically, it had a very low success rate, and there was the idea that clones were imperfect, that they were not as healthy or weren't able to live as long. And a lot of that comes from the fact that Dolly the sheep herself, she did live a shortened life. She did not live a full life like a normal sheep would have. As the technology improves for cloning, however, that's become less and less of a concern. You also have to think about biodiversity. We can't just clone one organism and repopulate the earth with it if we're talking about an endangered species, because you need that genetic diversity in a population. A population is healthiest if there is biodiversity or genetic diversity within it. Our next topic is genetic engineering, and this is going to be anytime you take DNA from one organism and you insert it into the DNA of a completely different kind of organism. This is done for the purpose of introducing some new trait to the organism, and that could be to make it grow faster or withstand frost or produce a new type of protein, depending on the application. So in genetic engineering, we basically are going to have to make recombinant DNA. No matter what type of organism we're using, in order to genetically engineer it, we need to give it DNA from an outside source. So a lot of times this is done with a bacterial plasmid, which is just that loop of DNA found in the cytoplasm. If there is a gene in this organism here, this is some kind of eukaryotic organism because it has a nucleus. If we take this gene out of the DNA of the eukaryotic cell, we can place it into this bacterial plasmid, and then when you insert that plasmid into the bacteria now contain these new instructions for making whatever that's a gene for. So we would now call that organism a transgenic organism. You may have heard it also called a genetically modified organism. That's the same thing, just a different way of saying it. A lot of times you hear transgenic organism when we're talking about animals and genetically modified organisms when we're talking about plants, but there's not really a functional difference between the meaning of those terms. So again, what we have done is we've taken foreign DNA, we have inserted it into the plasmid, we made recombinant DNA, and now this plasmid is inserted into bacterial cells. And those bacterial cells will now produce whatever that gene tells them to produce. And we can collect that gene product, it's probably some kind of protein, we can purify that, and we can then sell that at a pharmacy for somebody who needs that particular protein. So an example of that would be E. coli that has been engineered to produce human insulin. That's actually how we get a lot of our insulin today. It's made by bacteria. By inserting that human insulin gene into the bacteria, they would then have the capability of producing human insulin that is fit for use by diabetics. We also have an example of a transgenic plant or a GMO plant called BT corn. You may have heard of this before. This is commonly referenced also in the AP Human class if you take that class. And this is corn that has a gene from bacteria in it. BT is a type of bacteria. And the gene that they take from the bacteria is kind of like a natural pesticide found in that bacteria. It resists the pests. And so if you take that gene from the bacteria, insert it into the plant will now have the ability to fend off pests that might be eating it. It's not actually putting chemical pesticide in the plant. It's just producing a protein that the insects do not like to eat, and therefore the insects, once they take one bite of the corn, they will leave it alone. A third example of genetic engineering would be in the creation of what we call knockout mice or knockout organisms. If we are knocking out a gene, that means we have edited that DNA so that we've either removed a gene or we've made it completely non-functional. And this is really helpful when you're studying genetic diseases or just diseases that have some genetic component to them. If you can disable a gene, you can see what the result is in the mice that are produced, and you can learn more about what that gene does in that particular disease. The picture that you see here, this mouse on the right is a normal mouse, and this one on the left is a gene knockout mouse that has a gene that is involved in obesity. It has been disabled in this mouse, and therefore you can see that that mouse is unable to control its weight like the mouse on the right. So this would be a really good model organism for trying to study factors relating to obesity. There are some concerns about genetic engineering. You may have heard debate over this in the past. People might be concerned about possible long-term health effects from eating genetically modified foods. We don't really know of any health effects at the moment, but then again, as we learn more about genetically modified foods, as they become more common in our food sources, there is still much that we can learn about that. And there's also the potential effects of genetically modified plants on ecosystems and the biodiversity of the naturally occurring plants. Do we want to genetically modify a plant that could somehow take over and outcompete the native plants? That's definitely an important consideration. There's also a question about, do we need to label food that has genetically modified ingredients in it? Some people think, yes, that's important for making informed decisions, but other people think that there's kind of a scare tactic here by making people concerned that there's a genetically modified organism in their food, even if that organism is completely safe to consume. All right, the next topic we're talking about is that of genomics. Genomics is the study of genes and their functions. So in order to study genomics, you basically have to know all about the entire genome of an organism. A genome is just all of the genetic information that that organism has. It's not just one gene or one chromosome, but the entire body of all of the genes present in an organism. We can do this now because of gene sequencing technology has become very common, very easy, very accessible. You can even have your own DNA sequenced through various gene sequencing companies that can tell you more about your ancestry or whether or not you have a predisposition for a certain disease. Gene sequencing is definitely a very common use of biotechnology because we do have the ability to tell the exact order of A's, T's, C's, and G's in a genome. And this information over here to the side is just kind of an interesting comparison in the genome sizes of different organisms. Some of them you might find kind of surprising. Humans have about 3 billion bases in their genome. But even something like a vanilla plant, you might expect that to have less DNA than a human because we tend to think of humans as being so complex. But the vanilla plant has more than twice the amount of DNA that you would find in a human. So this is just an interesting comparison. It's certainly not any information that you have to memorize. The biggest example of genomics is the Human Genome Project. The Human Genome Project was a project to actually sequence the entire human genome. And from this project, which is now complete, we know the location of the somewhere around 20 to 25,000 different genes that are present in the human genome. And this is really important when you are studying medicine because if somebody has a genetic disease, you can kind of track down what gene is involved, where is that gene located, and that gives scientists a route to try to discover new treatments or cures for the disease, manufacture proteins that might help alleviate symptoms of a disease, and all of this comes from just our knowledge of where those genes are located. We don't quite know what all of them do yet. That's a lot of genes to figure out. But we do have them all mapped out onto the human chromosomes. Our next application of biotechnology is gene therapy. This is a really promising field of medicine where you can actually replace a defective gene and essentially cure a genetic disease. So the way this works is if there's a disease that is just involving one damaged gene, you could potentially insert a corrected version of that gene into the person with the disease using some kind of a vector like a virus. You're familiar with viruses, you know what they do. They infect living cells. Well, if we can modify the virus, instead of infecting it with the DNA that would make you sick, if it's making you well, if you are inserting DNA that's the correct version of a gene, then that could be very beneficial. Now, there are challenges here. It's difficult to overcome the immune system. Your body doesn't recognize that this virus has good DNA in it. It just knows it's a virus, and so it tries to attack it. And also, if there's already tissue damage, it's not really going to reverse that. It would just prevent more from occurring. So a great example here is cystic fibrosis. Cystic fibrosis, it involves a single gene for one membrane protein. It's called the CFTR. It's a transport protein in the membrane. When it doesn't work correctly, it can't transport the salts across the membrane, and that puts the water balance out of whack and it makes the mucus really thick and sticky. So to treat this with gene therapy, you would engineer a virus to have a corrected version of the gene for that CFTR protein. You would infect the person. It would need to go into their airways, needs to go into the lung tissue where the damage has occurred. And then the virus can inject the DNA into the lung cells of the person with cystic fibrosis. Once that gene is in there, that gene can be used to make the correct protein, and then any of the new cells produced in the lungs of this person would now have the correct version of the protein, and therefore they would not be producing that thick mucus. All right, last up we have stem cells. So these stem cells are going to be actually in chapter 5 of your textbook if you're looking up this information. And the types of stem cells we're going to talk about are either embryonic stem cells or adult stem cells. Everybody started out with embryonic stem cells, but after you got past the embryo stage when you were developing, those stem cells differentiated into a specific cell type, and they are no longer present in your body. The adult stem cells, however, are present in everybody at this exact moment. We'll talk about some examples of adult stem cells on one of the next couple slides. So before we do that, let's talk about why embryonic stem cells are so important. They're found only in the very earliest stages of an embryo, and so in order to do any kind of research with them, you would unfortunately have to destroy a human embryo to get those cells. Obviously, there's a lot of ethical questions and concerns that deal with that. Even if your end goal is something good, you do have to weigh that against the fact that you are destroying a human embryo. And just as an aside here, when people think of that, they tend to think of embryos from abortions, but that's not really the source of these embryos. We're talking mostly embryos from fertility clinics that are much earlier in the stages of development, in this very early stage here where it's only a few cells. Embryonic stem cells are really powerful because they have unlimited potential to turn into any type of cell in the body, whether we're talking about cells that make up your liver, pancreas, intestines, even neurons. Neurons are very famous for not being able to reproduce themselves, but they can be produced from embryonic stem cells. Adult stem cells, on the other hand, these are found in everyone. They do have the ability to differentiate, but they are much more limited in that ability. So in your bones, for example, you have bone marrow. Those are adult stem cells. And what they can do is they can turn into various types of blood cells, either red blood cells or the different types of white blood cells, those cells of your immune system that circulate in your blood. Those all come from these adult stem cells here. But these stem cells in your bone marrow couldn't turn into something like a neuron. They don't have that same potential as an embryonic stem cell. So what scientists are trying to do is they're trying to merge the best of both worlds and figure out a way to take actual cells from an adult and manipulate them in the lab so that they act like embryonic stem cells but didn't actually come from an embryo. And that's exactly what these induced pluripotent stem cells are. They're called iPSCs, and they are adult cells taken from an actual person. You don't have to kill the person to get them. You can just harvest some cells, treat them with certain chemicals in the lab to reprogram them, and then those iPSCs can turn into all of these cell types, even neurons, like in your brain, or motor neurons that would move your muscles. So if you have a brain injury or a spinal cord injury, this is a really promising way to produce new cells that might allow you to heal that energy. So that's going to be it for our applications of biotechnology. I know that was a really quick intro, but if you have any questions, please make sure you ask those in class.